remember pathogens, if it builds memory cells that last for decades, then why do you still get sick? Why do you catch a cold every winter? Why does the flu come back year after year? The answer lies not in any failure of your immune system, but in the relentless, never-ending evolution of the pathogens themselves. There is a common version of the immune system story that goes something like this. You encounter a pathogen, you get sick. Your immune system learns the pathogen, and now you're immune. Problem solved. Move on. If that were the whole story, nobody would ever get the flu twice. Nobody would catch multiple colds in a single winter. And we would have eliminated most infectious diseases centuries ago. The reason that story is incomplete is that pathogens evolve, and they evolve much, much faster than we do. Consider this. A human generation is roughly 25 to 30 years. In that time, we pass our genes to the next generation once. A bacterium like E. coli can divide every 20 minutes under ideal conditions. In a single human day, that bacterium can go through over 70 generations. In the time it takes for one human generation, bacteria have cycled through hundreds of thousands of generations. And every generation is an opportunity for mutation, for random changes in the pathogen's DNA that might, by chance, help it evade the immune system. Viruses evolve even faster. RNA viruses, which include influenza, HIV, and the common cold coronaviruses, are particularly prone to mutation because the enzyme they use to copy their RNA makes errors at a much higher rate than the enzymes that copy DNA. Every time a virus replicates inside one of your cells, the copies it produces are slightly different from the original. Most of those differences are meaningless or harmful to the virus. But occasionally, a mutation changes the shape of a surface protein, the very protein that your antibodies were designed to recognize. And when that happens, your existing antibodies no longer fit. The key has changed shape, and the lock you built for it doesn't work anymore. As far as your immune system is concerned, this mutated virus is a brand new threat. Your memory cells don't recognize it. You have to mount a primary response from scratch. This is why influenza comes back every year. The flu virus mutates its surface proteins, particularly two proteins called hemagglutinin and neuraminidase, just enough that last year's antibodies don't fully recognize this year's virus. Your immune system has excellent memory of last year's flu, but this year's flu is wearing a slightly different disguise, so you get sick again. This process is called antigenic drift, and it's a continuous, gradual change in the virus's surface proteins. It's the reason the flu vaccine needs to be updated every year. Scientists have to predict which strains will be circulating in the coming season and formulate the vaccine accordingly. Sometimes they predict accurately, and the vaccine is highly effective. Sometimes the virus changes in unexpected ways, and the vaccine is less effective. But even a partially matched vaccine usually reduces the severity of illness, because the antibodies it produces may not perfectly recognize the virus, but can still bind well enough to slow it down. There's an even more dramatic version of this called antigenic shift, which happens when two different flu viruses infect the same animal, usually a pig, which can be infected by both bird flu and human flu viruses, and their genetic material gets mixed together. The result is a flu virus with surface proteins that are completely different from anything the human immune system has seen before. No one has memory cells for this new combination. Everyone is vulnerable. This is how pandemic flu strains emerge. The 1918 Spanish flu, the 1957 Asian flu, the 1968 Hong Kong flu, the 2009 H1N1 pandemic. All of them were caused by antigenic shift. HIV takes immune evasion to another level entirely. HIV mutates so rapidly that a single infected person can harbor thousands of slightly different HIV variants simultaneously. By the time the immune system produces antibodies against one variant, the virus has already mutated into a form those antibodies can't recognize. It's like trying to hit a target that is changing shape faster than you can aim. This is the main reason we still don't have a highly effective HIV vaccine, despite decades of research and billions of dollars in funding.
The tuberculosis bacterium has a different strategy. Instead of mutating to avoid recognition, it hides. It infects macrophages, the very cells that are supposed to eat and destroy it, and then prevents the macrophage from killing it. The bacterium essentially sets up a safe house inside the macrophage, reproducing slowly and evading the immune response for years or even decades. An estimated 1/4 of the world's population is infected with tuberculosis bacteria that are sitting dormant inside macrophages, held in check by the immune system but not eliminated. If the person's immune system ever weakens through HIV infection, malnutrition, aging, or immunosuppressive medication, the tuberculosis can reactivate and cause active disease. Streptococcus pneumoniae, a common cause of bacterial pneumonia, has yet another trick. It has a thick carbohydrate capsule on its outer surface that makes it very difficult for phagocytes to grab and eat. Antibodies can bind to the capsule and help phagocytes get a grip. But here's the catch. There are over 90 different variants of the capsule, each with a slightly different structure. Antibodies against one variant don't recognize the others. The vaccine against Streptococcus pneumoniae includes 23 of the most common capsule variants, and even then, it only covers about 90% of disease-causing strains. The remaining 10% are wearing a capsule type that the vaccine doesn't target. The common cold is caused by over 200 different viruses: rhinoviruses, coronaviruses, adenoviruses, and others, and each one has its own set of surface antigens. You can be immune to one rhinovirus and completely vulnerable to the next because they look different to your immune system. This is why children get so many colds. They're encountering these viruses for the first time, one by one, building up an immunological library.
Adults get fewer colds, not because their immune systems are stronger in any absolute sense, but because they've already encountered and built memory against many of the viruses circulating in their community. There is also a phenomenon that certain viruses exploit, called immune evasion at the cellular level. Some viruses, cytomegalovirus, or CMV, is a classic example, have evolved proteins that specifically interfere with the MHC class I display system. Remember, MHC class I molecules show fragments of whatever the cell is making on the cell surface. If a cell is infected with a virus, viral protein fragments should appear on MHC class I, alerting killer T cells. But CMV produces proteins that block this process. It prevents the MHC class I molecules from reaching the cell's surface, so the infected cell looks normal to killer T cells. The virus is hiding inside the cell, producing its own proteins, but the cell's "I've been infected" sign has been turned off. This is where NK cells come back into the story. Remember that NK cells check for the presence of MHC class I molecules, and they kill cells that are missing them. So when CMV shuts down MHC class I, it avoids killer T cells, but it exposes itself to NK cells. The virus has solved one problem and created another. This is the arms race in miniature. Every evasion strategy has a counter, and every counter has a counter-counter. The point of all this is that your immune system faces a constantly evolving enemy. The pathogens you encounter today are not the same pathogens your grandparents encountered. They have been shaped by millions of years of evolutionary pressure from immune systems exactly like yours, and they have developed an astonishing array of strategies to survive. And your immune system has responded by developing its own array of counter-strategies, the layered, multi-tiered, redundant system we've been describing throughout this video. Barriers, mucus, complement,
Macrophages, neutrophils, NK cells, dendritic cells, T cells, B cells, antibodies, memory cells, each one compensating for the other's weaknesses, each one adding another layer of protection that a pathogen has to overcome. It is an arms race without end, and neither side is winning. The pathogens keep evolving new tricks, and the immune system keeps evolving new defenses. The result is a kind of dynamic equilibrium. You get sick sometimes, but not often, and usually not seriously. The immune system doesn't eliminate all threats, but it keeps them under control, most of the time. But there is one threat that doesn't come from outside. It doesn't evolve in the wild. It doesn't mutate to evade your antibodies. It comes from within your own body, from cells that were once perfectly normal, perfectly healthy, and perfectly loyal, until they weren't. And your immune system's ability to deal with this threat may be the most important thing it does. Every day, your body produces somewhere in the neighborhood of 300 billion new cells. Every single one of those cell divisions requires the complete, accurate copying of your entire genome, roughly 3 billion base pairs of DNA. And despite the extraordinary precision of the enzymes that copy DNA, mistakes happen. Mutations. A base pair gets swapped, a segment gets deleted, a gene gets duplicated. The estimates vary, but it's generally thought that each cell division introduces roughly one new mutation on average, sometimes none, sometimes a few. The vast majority of these mutations are harmless. They occur in parts of the genome that don't code for anything important, or they change a protein in a way that doesn't affect its function. These mutations are biological background noise. They accumulate over your lifetime, but they don't cause any problems. But occasionally, a mutation lands in exactly the wrong place. It activates a gene that promotes cell growth when it should be turned off. Or it deactivates a gene that's supposed to put the brakes on cell division. And when this happens, the cell begins to behave abnormally. It starts dividing when it shouldn't. It ignores the signals from neighboring cells that tell it to stop. It becomes, in a very real sense, selfish, prioritizing its own reproduction over the needs of the organism as a whole. This is the beginning of cancer. And it happens far more often than most people realize. The current scientific understanding is that your body produces hundreds of potentially cancerous cells every single day. Cells with mutations in growth-controlling genes. Cells that have taken the first steps toward becoming tumors. And your immune system kills most of them. This process is called immunosurveillance. NK cells, killer T cells, and macrophages are constantly patrolling the body, checking cells for signs of abnormality. A cell that has accumulated too many mutations often begins to display unusual molecules on its surface, stress markers that NK cells can detect, or abnormal proteins that killer T cells can recognize through MHC class 1. When the immune system identifies a cell as abnormal, it destroys it, usually through the same mechanisms it uses to kill virus-infected cells. Perforins, granzymes, apoptosis. The cancerous cell is eliminated before it ever has the chance to form a tumor. We know immunosurveillance is real because of what happens when it fails.
People with suppressed immune systems, organ transplant recipients on immunosuppressive drugs, people with advanced HIV, have significantly higher rates of certain cancers. Without the immune system's constant patrol, the potentially cancerous cells that would normally be caught and destroyed have a much better chance of surviving and multiplying. But cancer cells, like pathogens, can evolve.
And some of them evolve in ways that allow them to evade the immune system. One of the most common evasion strategies is the same one we saw with viruses, reducing MHC class I expression. A cancer cell that stops displaying MHC class I molecules on its surface becomes invisible to killer T cells, because killer T cells need to see antigens presented on MHC class I to recognize a target. The cancer cell has, in effect, taken off its name tag. But again, this exposes it to NK cells, which kill cells that are missing MHC class I. So some cancer cells find a middle ground. They display enough MHC class I to avoid NK cell killing, but not enough to trigger a strong killer T cell response. They're walking a tightrope between two different surveillance systems. Another evasion strategy involves a molecule called PDL1. This molecule, when displayed on a cell's surface, sends a stand-down signal to killer T cells that come into contact with it. PDL1 essentially says, "I'm fine, don't kill me." Normal, healthy cells use PDL1 to prevent unnecessary immune attacks. But some cancer cells have figured out how to overexpress PDL1 to produce huge amounts of it on their surfaces, broadcasting a constant "don't kill me" signal to every killer T cell in the area. The T cells arrive, see the PDL1, and back off. The cancer cell is left alone to divide and grow. This discovery led to one of the most significant breakthroughs in cancer treatment in recent decades: immune checkpoint inhibitors. These are antibodies designed to block PDL1, or its receptor on T cells, PD1, and prevent the cancer cell from sending the stand-down signal. When the checkpoint is blocked, killer T cells can recognize the cancer cell for what it is and destroy it. Checkpoint inhibitors have shown remarkable results in certain cancers: melanoma, lung cancer, kidney cancer, extending survival in some patients by years.
But checkpoint inhibitors work by removing a safety mechanism from the immune system. By blocking the stand-down signal, they allow killer T cells to be more aggressive, not just against cancer cells, but potentially against healthy tissue too. The side effects of checkpoint inhibitors can include autoimmune-like symptoms: inflammation of the gut, the liver, the lungs, the skin. It's the same fundamental trade-off we've seen throughout this entire discussion. The immune system's power is a double-edged sword, and any attempt to unleash it carries the risk of friendly fire. There are other approaches to cancer immunotherapy that are even more targeted. One of the most exciting is called CAR-T cell therapy. In this approach, doctors take T cells from the patient's own blood, genetically engineer them in the laboratory to express a receptor that specifically recognizes a molecule on the patient's cancer cells, multiply those engineered T cells in huge numbers, and then infuse them back into the patient. The engineered T cells, the CAR T cells, then seek out and destroy the cancer cells with extraordinary precision. CAR T-cell therapy has produced remarkable results in certain blood cancers, particularly certain types of leukemia and lymphoma. Some patients who had exhausted all other treatment options have gone into complete remission after CAR-T therapy. But it's still a new and expensive technology, and it doesn't work for all cancer types. Solid tumors, the kind that form lumps in the breast, the lung, the colon, are much harder for T cells to penetrate than blood cancers, because solid tumors create a microenvironment that suppresses immune cell function.
There's another approach being developed at cancer research centers around the world: personalized cancer vaccines. The idea is to analyze the mutations in a patient's specific tumor, identify which mutations produce abnormal proteins that the immune system could potentially recognize, and then create a vaccine that trains the immune system to attack cells displaying those proteins. Unlike traditional vaccines, which are designed for a specific pathogen and given to millions of people, a personalized cancer vaccine is designed for one patient's specific tumor. It's a bespoke immune response, custom-built for one person's cancer. This is still largely experimental. Clinical trials are underway, and some early results are promising, but we are at the very beginning of this approach. The complexity is enormous. Every patient's tumor is genetically unique, so every vaccine has to be designed from scratch. But the principle is sound. The immune system has the tools to kill cancer cells. It just needs to be pointed in the right direction. The broader point here is that the relationship between the immune system and cancer is a constant back and forth of surveillance and evasion, playing out over years. Your immune system is killing precancerous cells every day. The cancers that actually develop into clinical disease are the ones that found a way past the surveillance, the ones that evolved evasion mechanisms fast enough to outrun the immune response. And understanding that relationship is transforming cancer treatment from a blunt force approach, chemotherapy that kills everything rapidly dividing, radiation that damages all cells in its path, into something far more precise. Therapies that harness the immune system's own targeting ability to seek and destroy cancer cells while leaving healthy tissue intact. We're not there yet, but the direction is clear and the progress is real.
Now, we've spent the last several chapters discussing the immune system's extraordinary capabilities. Its ability to recognize pathogens, to build specific weapons against them, to remember past infections, to detect cancer cells, and to mount responses that range from the brutally fast to the surgically precise. But we haven't yet addressed a question that is probably on your mind, because it's a question that affects your daily life in ways that immunology textbooks rarely discuss. What actually determines how strong your immune system is? What makes one person catch every cold that goes around, while another person seems to never get sick? Is it genetics, diet, exercise, sleep, stress? The answer, it turns out, is all of the above. There was a study conducted at the Pasteur Institute in Paris, one of the most prestigious immunology research centers in the world, that looked at this question directly.
The researchers recruited 1,000 healthy adults between the ages of 20 and 70, half men and half women. They collected blood samples from each person and exposed those samples to various pathogens in the laboratory, measuring how strongly each person's immune cells responded. They also collected detailed information about each person's genetics, lifestyle, diet, smoking habits, and medical history. After several years of analysis, the results identified 5 factors that had the greatest influence on how well a person's immune cells functioned: genetics, age, biological sex, smoking status, and infection with cytomegalovirus. Let's go through each of these. Genetics sets the foundation. Your DNA determines the types of MHC molecules you produce, the range of T cell and B cell receptors you can generate, the intensity of your inflammatory responses, the efficiency of your complement system, and dozens of other immune parameters.
Some of this genetic variation is protective. Certain MHC types are associated with better outcomes against specific infections. Some of it is disadvantageous. Certain genetic profiles make a person more susceptible to autoimmune diseases or less responsive to vaccines. But the key point is that genetic variation in immune function comes down to trade-offs, not a simple scale of strong versus weak. A genetic profile that makes you more resistant to one type of infection might make you more susceptible to another, or more prone to autoimmunity. The immune system is not optimized for any single threat. It's a compromise, balancing protection against a vast range of possible dangers. Age is the second major factor, and we've already discussed why. The thymus shrinks throughout life, reducing the production of new T cells. The overall diversity of the T cell repertoire declines. The ability to mount a strong primary response to a genuinely new pathogen decreases. Older adults also tend to have higher levels of chronic, low-grade inflammation, a phenomenon sometimes called inflammaging, which can paradoxically weaken immune responses by keeping the system in a constant state of mild activation. The immune system in an older adult is like an army that has fought many battles and remembers all of them, but has fewer fresh recruits and less energy for new campaigns. Biological sex matters more than most people realize. Epidemiological data consistently shows that men are, on average, more susceptible to infectious diseases than women, while women are more prone to autoimmune diseases. The reasons are complex and not fully understood, but they likely involve sex hormones. Estrogen tends to enhance immune responses, while testosterone tends to suppress them. And genetic factors, since many immune-related genes are located on the X chromosome.
Women have two X chromosomes and therefore a double dose of those genes, which may contribute to stronger immune responses, but also a greater risk of the immune system overreacting against self-tissues. Smoking is a direct, dose-dependent immunosuppressant. Cigarette smoke damages the cilia lining the respiratory tract, the mucociliary escalator that we discussed earlier, reducing the body's ability to clear pathogens from the airways. It impairs the function of macrophages and neutrophils in the lungs. It increases chronic inflammation in the respiratory system, while simultaneously reducing the effectiveness of adaptive immune responses. Smokers get more respiratory infections, recover more slowly, and respond less well to vaccines than non-smokers. If there is one lifestyle factor that most directly and unambiguously weakens the immune system, it is smoking. Cytomegalovirus, CMV, is a fascinating case. CMV is a very common virus. Roughly 50% of adults in developed countries and an even higher percentage in developing countries are chronically infected. In healthy people, CMV usually causes no symptoms at all. The immune system keeps it under control, but never fully eliminates it. The virus persists in a latent state, hiding inside cells for the rest of the person's life. This chronic, lifelong infection reshapes the immune system over time. A significant fraction of the body's T cells become dedicated to keeping CMV suppressed, which means fewer T cells are available for other tasks. In older adults, this CMV-driven reallocation of T cell resources may contribute to the overall decline in immune function with age. Beyond these 5 major factors, there are several others that play important roles, even if they are harder to quantify precisely. Sleep is critical. During sleep, the body produces higher levels of certain cytokines that promote immune function. Sleep deprivation, even short-term, has been shown to reduce the production of antibodies after vaccination and to increase susceptibility to infection. In one well-known study, researchers exposed healthy volunteers to the common cold virus and tracked who got sick.
The participants who had slept fewer than 7 hours per night in the week before exposure were nearly 3 times more likely to develop a cold than those who had slept 8 hours or more. Exercise has a complicated relationship with immunity. Moderate, regular exercise, the kind that most health guidelines recommend, around 30 minutes of brisk activity most days, is consistently associated with better immune function. People who exercise regularly get fewer upper respiratory infections, respond better to vaccines, and have lower levels of chronic inflammation.
The mechanisms are not fully understood, but exercise increases circulation, which helps immune cells move through the body more efficiently, and it may help flush bacteria out of the lungs and airways. But extreme exercise, the kind that marathon runners and professional athletes engage in, can temporarily suppress immune function. After a marathon or an intense training session, there is a window of several hours to a couple of days, during which the athlete's immune defenses are reduced. This is sometimes called the open window hypothesis, and it's the reason that elite athletes often report getting sick after major competitions or training peaks. The immune system seems to have a sweet spot. Moderate exertion strengthens it, but exhaustive exertion temporarily weakens it. Nutrition matters, but not in the way that supplement advertisements would have you believe. Severe malnutrition, insufficient calories or insufficient protein, unambiguously weakens the immune system.
This is well established and is one of the reasons that infectious diseases are so much more deadly in populations suffering from famine or extreme poverty. Certain micronutrient deficiencies also impair immune function. Vitamin D, vitamin C, zinc, and iron are all necessary for various immune processes, and deficiency in any of them can reduce the body's ability to fight infections. But here's the thing. If you're eating a normal, reasonably varied diet, you're almost certainly getting enough of all of these nutrients. The evidence that taking extra vitamins or supplements, beyond what you get from food, provides any additional immune benefit is weak to non-existent. There is no pill you can take that will boost a normally functioning immune system. The supplement industry is worth billions of dollars and sells the idea that you can buy better immunity, but the science simply does not support that claim for people who are not malnourished. What does have good evidence behind it is the role of the gut microbiome. Your intestines host roughly 40 trillion bacteria, more bacterial cells than human cells in your entire body. And these bacteria play a critical role in immune function. Approximately 70% of all immune cells in your body are located in and around the gut, precisely because the gut is the site where the most foreign material enters the body. Everything you eat carries bacteria, and the immune system in the gut has to make constant decisions about what is a threat and what is not. The bacteria living in your gut, collectively called the gut microbiome, are not enemies. They are partners. They help digest food. They produce vitamins. They produce short-chain fatty acids that nourish the cells lining the gut. And critically, they train the immune system. The constant, low-level interaction between gut bacteria and immune cells teaches the immune system to distinguish between harmless microorganisms and dangerous ones. A diverse, healthy microbiome is associated with a well-calibrated immune system, one that responds vigorously to real threats but doesn't overreact to harmless substances. An impoverished microbiome, one with reduced diversity, often caused by a diet high in processed food and low in fiber, or by repeated courses of antibiotics, is associated with a less well-calibrated immune system. There's growing evidence that reduced microbial diversity is linked to higher rates of allergies, autoimmune diseases, and inflammatory conditions. This is related to what's sometimes called the hygiene hypothesis, or more accurately, the old friends hypothesis, which proposes that modern humans living in cleaner environments with less microbial exposure than our ancestors have immune systems that are undertrained and prone to overreaction. Children who grow up on farms, exposed to a wide variety of environmental microorganisms, have lower rates of asthma and allergies than children raised in sterile urban environments. Children who attend daycare and are exposed to other children's germs early in life tend to get sick more in the first couple of years, but have lower rates of allergies later. The immune system needs exposure to a diverse array of microorganisms to calibrate itself properly, and the modern world, with its hand sanitizer, antibiotics, processed food, and limited outdoor play, may not be providing that exposure in sufficient quantities. This doesn't mean that hygiene is bad or that you should stop washing your hands. Hygiene saves lives. Clean water and sanitation are among the greatest public health achievements in history. But there may be a balance between too much microbial exposure, which leads to infectious disease, and too little, which leads to allergies and autoimmunity. Finding that balance is one of the great challenges of modern immunology. Stress is the final factor we should discuss, and it is perhaps the most underestimated. Chronic stress, the kind that comes from ongoing life challenges, caregiving responsibilities, financial insecurity, or demanding work environments sustained over months or years, has a measurable negative effect on immune function. The mechanism involves the stress hormone cortisol, which is produced by the adrenal glands in response to perceived threats. In the short term, cortisol prepares the body for a fight or flight response, which actually includes some immune enhancement, making sure that if you're injured in the fight, your immune system is ready to deal with wound infections. But when stress becomes chronic and cortisol levels remain elevated for weeks or months, the effect reverses. Chronic cortisol exposure suppresses the adaptive immune system. It reduces the production and activity of T cells and B cells. It impairs the ability to mount effective antibody responses to vaccines. It increases susceptibility to viral infections. In one study, caregivers of family members with Alzheimer's disease, people under sustained long-term psychological stress, had significantly reduced immune function compared to age-matched controls. They took longer to heal from wounds, responded more weakly to flu vaccines, and had higher levels of inflammatory markers in their blood. There is also emerging evidence that acute psychological stress can affect immune function in the very short term. Research using virtual reality to induce stress responses in healthy volunteers found that even 30 minutes of acute stress caused measurable changes in immune cell distribution. Specifically, a drop in the number of B cells and T cells in the blood and an increase in neutrophils. The adaptive immune cells were retreating into the bone marrow, while the innate immune cells were being mobilized. This makes evolutionary sense. If you're facing an immediate physical threat, you need the fast, violent, innate response more than the slow, specific adaptive response. Neutrophils can handle a wound right now. T cells can wait. But this short-term redistribution, if sustained by chronic stress, becomes a liability. The adaptive immune system is chronically suppressed, leaving the person more vulnerable to viral infections and less able to respond to vaccines. The practical implication is clear, even if it's not easy to act on. Managing stress is an immune health issue, not just a mental health issue. The mind and the immune system are not separate systems operating independently. They are connected through hormones, through the nervous system, specifically the vagus nerve, which directly innervates many immune organs, and through mechanisms we are only beginning to understand.
The placebo effect, the well-documented phenomenon in which patients improve simply because they believe they are receiving treatment, almost certainly involves real changes in immune function, triggered by psychological expectation. Clinical trials of immune-related therapies must use placebo controls, precisely because the psychological state of the patient can genuinely alter the course of the disease. So let me draw all of this together into a practical picture.
Your immune system's effectiveness at any given moment is the product of your genetics, your age, your biological sex, your infection history, your sleep, your exercise habits, your diet, your gut microbiome, your stress level, and your vaccination status. Some of these you can't control, many of them you can. And the thing that sits above all of these, the one intervention that has the clearest, strongest, most unambiguous evidence of improving immune outcomes, is the one we've already discussed: vaccination. Vaccines work. They are the single most effective tool humanity has ever developed for enhancing immune function against specific threats. Getting vaccinated gives your adaptive immune system the precise information it needs to protect you from a specific pathogen before you ever encounter it, something no supplement or lifestyle change can replicate. But vaccination is also a story of limitations, as we discussed.
We have excellent vaccines for some diseases and no vaccines at all for others. We have vaccines that provide lifelong protection and vaccines that need to be updated every year. The future of vaccinology is about closing those gaps, developing better vaccines against the pathogens that have so far evaded our best efforts. And it's about something else too, something that goes beyond fighting infections, because the immune system, as we've seen, does far more than defend against external threats. It maintains the body's internal order, killing cancer cells before they become tumors, cleaning up damaged tissue, resolving inflammation after an injury, keeping the trillions of gut bacteria in check, preventing the body from attacking itself. The immune system functions more like an ecosystem than an army. And like any ecosystem, it depends on balance. And when I say balance, I mean something very specific. Consider what we've covered so far.
The innate immune system is powerful, but indiscriminate. Neutrophils dump bleach on bacterial infections, and some of your own cells get damaged in the process. The adaptive immune system is precise, but potentially self-destructive. Killer T cells can eliminate virus-infected cells one by one, but if they target the wrong cells, you get autoimmune disease. Complement proteins punch holes in bacteria with lethal efficiency, but they need to be kept in check or they'll damage your own tissues. Inflammation brings immune cells and proteins to the site of infection, which is essential. But chronic inflammation causes tissue damage, contributes to cardiovascular disease, and may accelerate neurodegeneration. Every single component of the immune system is a trade-off between power and control, between aggression and restraint, between speed and precision. The system works because all of these trade-offs are carefully balanced. The inflammatory response ramps up when it's needed and ramps down when the threat is cleared. The killer T cells are activated by co-stimulatory signals and suppressed by regulatory T cells. The complement cascade is held in check by regulatory proteins on the surface of your own cells that prevent complement from attacking self-tissue. When any part of this balance is disrupted, when inflammation doesn't resolve, when regulatory T cells fail, when complement regulation breaks down, disease follows. Sometimes the disruption is genetic. Sometimes it's environmental. Sometimes it's a combination of both. but the underlying principle is always the same. The immune system's greatest strength is also its greatest vulnerability, and the line between protection and destruction is thinner than most people realize. So what tips that balance one way or the other?
What determines whether your immune system is a well-calibrated defense network or a ticking time bomb? We've talked about what happens when the system is too weak. Infections overwhelm it. We've talked about what happens when it's too aggressive. Autoimmune disease. But there is a third scenario, one that every person watching this has experienced personally, and it raises a question we've been circling around since the beginning of this video. When you catch a cold, your immune system launches a full-scale defense. Macrophages, neutrophils, cytokines, inflammation, interferons, and eventually T cells and antibodies, the whole cascade we've been describing. Your immune system wins. It always wins against a cold. And yet, for about a week, you feel absolutely terrible. Your nose is running, you're coughing, you have a fever, your entire body aches. If the immune system is winning, why does it feel like you're losing? The answer to that question is one of the most counterintuitive things in all of medicine. When you're sick, when you have a cold or the flu, you feel awful. You have a fever, your nose is running, your throat hurts, your muscles ache, you're exhausted. Every instinct tells you to lie in bed, drink soup, and wait for it to pass. And here is the thing that most people don't realize. Almost none of those symptoms are caused by the pathogen. Almost all of them are caused by your own immune system. Let me say that again because it's important. When you have a cold, the virus itself is doing remarkably little direct damage to your body. It's infecting cells in your nasal passages and your throat, sure, and those cells are dying. But the runny nose, the sore throat, the fever, the aches, the fatigue, those are all the result of your immune system's response to the virus, not the virus itself.
Let's start with fever, because fever is one of the most commonly misunderstood immune responses. When macrophages detect a pathogen and start releasing cytokines, some of those cytokines, called pyrogens, enter the bloodstream and travel to the hypothalamus, the part of the brain that acts as the body's thermostat. The pyrogens signal the hypothalamus to raise the body's temperature set point. The hypothalamus responds by triggering the mechanisms that increase body temperature: shivering, which generates heat through muscle contraction, constriction of blood vessels in the skin, which reduces heat loss, and behavioral changes, the urge to bundle up under blankets, curl into a ball, drink warm liquids. The result is a fever. And here's the key thing to understand. Fever is a deliberate, controlled, purposeful response with several specific benefits. First, a moderate fever, around 100 to 102 degrees Fahrenheit, speeds up the metabolic rate of your immune cells. Chemical reactions run faster at higher temperatures, and immune cells are no exception. Macrophages phagocytose bacteria more quickly. T cells divide faster. Antibodies are produced at a higher rate. The entire immune response is accelerated. Second, fever makes the environment less hospitable for many pathogens. Most bacteria and viruses are optimized to replicate at normal human body temperature, 98.6 degrees Fahrenheit. A few degrees above that, and their replication slows. They don't function as well. Fever creates an environment where the pathogen is struggling and the immune cells are working faster. It tilts the playing field in your favor. Third, fever triggers the liver and spleen to sequester iron and zinc from the bloodstream. Iron and zinc are essential nutrients for bacterial growth. By pulling these minerals out of circulation, the body starves bacteria of the raw materials they need to reproduce. This is called nutritional immunity, and it's an ancient defense mechanism shared by virtually all animals. So fever helps your immune system work faster, slows down pathogen replication, and starves bacteria of essential nutrients. It is, by every measure, a beneficial response. And yet, what does almost every person do when they get a fever? They take a fever reducer, acetaminophen, ibuprofen. They bring the temperature back down to normal. Now, I want to be careful here because this is an area where the nuance matters. There are situations where fever reduction is appropriate and even necessary. A fever above roughly 104 degrees Fahrenheit can be dangerous, particularly in young children, because at very high temperatures, proteins begin to denature. They lose their shape and stop functioning. Enzymes, which are proteins, are particularly sensitive, and the body's enzymatic machinery can begin to fail at extreme temperatures. High fevers can also cause febrile seizures in young children, which are frightening, though usually not dangerous. In these cases, bringing the fever down is the right medical decision. But for a moderate fever, the kind you get with a typical cold or flu, where your temperature might reach 100 or 101, there is a reasonable argument that taking a fever reducer, while it makes you feel better in the short term, may actually prolong the illness by removing one of your immune system's most effective tools. Studies on this question have produced mixed results, but some have found that treating fever with antipyretics is associated with slightly longer duration of symptoms. You feel better sooner, but you may stay sick longer. The aches and fatigue you feel during an illness are also deliberate. Cytokines released during the immune response, particularly interleukin-1 and tumor necrosis factor, act on the brain to produce what immunologists call sickness behavior. This includes fatigue, loss of appetite, increased sensitivity to pain, social withdrawal, and sleepiness. All of these behaviors serve an evolutionary purpose. They force you to rest. By making you feel too tired and too achy to go about your normal activities, the immune system ensures that your body's energy resources are directed toward fighting the infection rather than being wasted on running around, working, socializing, and doing other things that can wait. Sickness behavior is the immune system hijacking your brain. It's commandeering your behavior for the good of the defense effort. You don't want to lie in bed. Your immune system wants you to lie in bed, because lying in bed means more energy is available for producing antibodies and T cells. Your runny nose is the result of inflammation in your nasal passages. The immune response to the virus causes blood vessels in the nasal lining to dilate and become more permeable, leading to fluid leakage into the nasal cavity. Mucus production ramps up. The cilia continue to beat, pushing the mucus, along with the virus particles trapped in it, out of your nose. Sneezing is a reflex triggered by irritation of the nasal passages, and it serves to forcefully expel mucus and pathogens. It's messy and uncomfortable, but it's functionally useful. Every sneeze expels millions of virus particles that would otherwise continue to replicate in your nasal tissue. Your sore throat is the result of inflammation in the pharynx. Immune cells are flooding into the tissue, releasing cytokines, killing infected cells and virus particles. The swelling and the chemical signals from the immune response stimulate pain receptors. You feel it as a raw, scratchy soreness. It's your immune system at work. Coughing similarly is a reflex designed to clear the airways of mucus, debris, and pathogens. When immune cells in the respiratory tract detect pathogens and initiate inflammation, the resulting mucus production and tissue swelling trigger the cough reflex. Like sneezing, coughing is uncomfortable but functional. It's your body physically ejecting infectious material from your lungs and airways. So the next time you're lying in bed with a cold, feeling utterly miserable, consider this. Your body is doing exactly what it's supposed to do. Every symptom you're experiencing is a weapon being deployed in your defense. The fever is cooking the pathogens and turbocharging your immune cells. The mucus is trapping the invaders. The coughing and sneezing are expelling them. The fatigue is forcing you to conserve energy for the battle. The aches are keeping you still. You are not sick because the virus is destroying your body. You are sick because your immune system is destroying the virus, and the battle generates collateral damage that you experience as symptoms. This understanding changes the way you think about illness, and it should change the way you think about treatment. The goal of treatment should generally be to support the immune response, not to suppress it. Rest, hydration, adequate nutrition, and sleep are the most effective things you can do when you're sick, because they support the immune system's work. Fever reducers and cough suppressants have their place, particularly when symptoms are severe enough to prevent sleep or cause significant distress, but they should be used thoughtfully, with an understanding of what you're suppressing. And this brings us to a part of your body that you probably don't think about very much, but that contains the single largest concentration of immune cells anywhere in your body: your gut. When you eat something, the food travels from your mouth through your esophagus into your stomach, where it's doused in hydrochloric acid and broken down by enzymes. From there, the partially digested food moves into the small intestine, where the majority of nutrient absorption takes place, and then into the large intestine, where water is reabsorbed and the remaining material is prepared for excretion. This journey, from mouth to exit, covers roughly 30 feet of internal tubing, and every inch of that tubing is open to the outside world. Your digestive tract is, technically, a long tube that runs through your body, and the inside of that tube is continuous with the outside environment. The food you eat brings with it bacteria, viruses, fungi, parasites, and an enormous variety of foreign proteins, many of which your immune system has never seen before. This makes the gut the single largest interface between your body and the outside world. More external material passes through your gut than through your lungs and skin combined. And the body has responded by placing approximately 70% of all immune cells in and around the intestine. The gut-associated lymphoid tissue, sometimes abbreviated GALT, is a vast network of immune cells, lymph nodes, and specialized structures like Peyer's patches, clusters of immune tissue embedded in the wall of the small intestine, that monitor everything passing through the digestive tract. The immune system in the gut has to make a decision about every single molecule that comes through. Is this food, or is this a threat? And this is where it gets complicated, because the gut also contains roughly 40 trillion bacteria, 40 trillion microorganisms living inside you all the time. They coat the walls of your intestine. They break down fiber that your own enzymes can't digest. They produce vitamins, vitamin K, some B vitamins, that your body absorbs and uses.
They produce short-chain fatty acids that nourish the cells lining your gut. They compete with pathogenic bacteria for space and resources, making it harder for dangerous organisms to gain a foothold. These bacteria, the gut microbiome, are not enemies. They are essential partners. You need them and they need you. The relationship is mutualistic. They get a warm, nutrient-rich environment to live in, and you get their metabolic byproducts and their competitive exclusion of pathogens. But they're still bacteria. They still have the molecular signatures that the immune system is designed to detect and respond to. Bacterial cell wall components, flagellin, foreign DNA. If these bacteria were anywhere else in your body, in your blood, in your lungs, in your liver, your immune system would attack them instantly. So how does the immune system tolerate 40 trillion bacteria in the gut without launching a constant inflammatory assault? The answer involves a sophisticated communication system between the gut bacteria and the immune cells that surround them. The gut microbiome trains the immune system from the moment of birth. When a baby is born, particularly through a natural vaginal delivery, it is colonized by bacteria from the mother's birth canal and gut. These bacteria begin interacting with the baby's immune system immediately, and those interactions shape how the immune system develops. The bacteria communicate with the gut immune cells through a variety of mechanisms. They produce metabolites that signal to immune cells, "I am a normal, expected resident. I am not a threat." The immune system learns to recognize these signals and to suppress inflammatory responses against the resident bacteria while maintaining readiness against genuinely dangerous pathogens. Regulatory T cells in the gut play a crucial role in this tolerance. They actively suppress immune responses against the microbiome, keeping the peace. When this communication breaks down, the consequences can be severe. Inflammatory bowel diseases, Crohn's disease and ulcerative colitis, are conditions in which the immune system in the gut loses its tolerance for the resident bacteria and begins attacking them. The result is chronic inflammation of the intestinal lining, leading to pain, diarrhea, bleeding, and tissue damage. These are autoimmune-like conditions, and they illustrate what happens when the carefully maintained truce between the immune system and the microbiome falls apart. The composition of your gut microbiome is shaped by many factors, but diet is one of the most important. A diet rich in fiber, vegetables, fruits, legumes, whole grains, promotes a diverse microbiome because fiber is the primary food source for many beneficial gut bacteria.
When they ferment fiber, they produce short-chain fatty acids like butyrate, which has anti-inflammatory properties and helps maintain the integrity of the gut lining. A diet low in fiber and high in processed foods and refined sugars tends to reduce microbial diversity, which is associated with poorer immune calibration and higher rates of inflammatory and autoimmune conditions. This is why nutritionists and immunologists alike tend to give the same dietary advice. Eat vegetables, eat fiber, eat a diverse range of plant-based foods. Not because any single food contains a magic immune-boosting compound, but because a diverse diet supports a diverse microbiome, and a diverse microbiome supports a well-calibrated immune system. The importance of early microbial exposure is hard to overstate. As we discussed earlier, children born by caesarean section tend to be colonized by different bacteria than children born vaginally. More skin bacteria, fewer gut and vaginal bacteria. And some studies suggest this is associated with slightly higher rates of allergies and immune-related conditions. Breastfed children receive antibodies and immune-modulating factors through breast milk, and they're exposed to bacteria on the mother's skin, further shaping their developing immune systems. Children who grow up in environments with high microbial diversity, on farms, with animals, with siblings, tend to have lower rates of asthma and allergies. There is a striking parallel here with language development. If you grow up hearing and speaking a language, you develop fluency naturally. Certain sounds, like the German ich sound, are easy to learn in childhood, but very difficult to acquire as an adult. The microbiome is similar. The microbial community you establish in early childhood shapes your immune development in ways that are difficult to change later. You can improve your microbiome as an adult by changing your diet, by reducing unnecessary antibiotic use, but it's harder than building a healthy microbiome from the start. The COVID-19 pandemic provided an unintentional experiment in this regard. During the period of mask wearing, social distancing, and enhanced hygiene, children had dramatically reduced exposure to common respiratory viruses, not just COVID, but also RSV, influenza, rhinoviruses, and others. When these measures were lifted, pediatric hospitals saw a surge in respiratory infections because 2 or 3 years' worth of children were encountering these viruses for the first time simultaneously. Their immune systems hadn't been trained on these pathogens during the normal developmental window, and they were playing catch-up all at once.
This doesn't mean that pandemic precautions were wrong. They saved millions of lives.
But it illustrates the principle that microbial exposure is not just inevitable, it's necessary. The immune system needs to encounter a diverse array of microorganisms during development to calibrate itself properly. Too little exposure, and the immune system may overreact to harmless stimuli, causing allergies, asthma, and autoimmune conditions. Too much exposure, or exposure to the wrong things, and you get infectious disease. Balance, again, is everything. The gut, then, is as much an immune organ as a digestive one. Arguably, the immune system's primary training ground, the place where the body learns to distinguish between the harmless and the harmful, between partner and pathogen. The 40 trillion bacteria living inside you are teachers, co-workers, and allies.
And the immune system's ability to maintain a functional relationship with them is one of the most important determinants of overall health. But there is one final chapter to this story. One more dimension of the immune system that ties everything together. The dimension that most directly answers the question we started with. You were dying, right now, while watching this video. And you didn't know it. What saved you? Let's go back to where we started. You're sitting here, watching this video. You've been sitting here for a while now. And during that time, your body has been doing things you were completely unaware of. In the lining of your nasal passages, mucus has been quietly trapping particles from the air you've been breathing. Dust, pollen, bacterial spores, viral particles. The cilia lining your airways have been beating rhythmically, pushing that mucus upward, carrying the trapped debris away from your lungs and toward your throat, where you've been unconsciously swallowing it. The particles that made it to your stomach were dissolved by hydrochloric acid. On the surface of your skin, antimicrobial peptides and the low pH of your sweat have been suppressing the growth of the bacteria that are always present, the normal skin flora. Every time you touched something, your phone, your keyboard, your face, you transferred bacteria from that surface to your skin and from your skin to that surface. Your skin handled all of it without involving a single immune cell. In your intestines, approximately 70% of your immune cells have been monitoring the passage of food and bacteria through your digestive tract. Macrophages and dendritic cells in the gut wall have been sampling the contents, checking for anything unusual. Regulatory T cells have been keeping the peace between your immune system and the trillions of bacteria that live there, A few bacteria that tried to cross the intestinal barrier were intercepted and destroyed by macrophages and complement proteins before they reached the bloodstream. Somewhere in your body, maybe in your skin, maybe in your liver, maybe in your lung, a cell underwent a mutation during its most recent division. The mutation activated a gene involved in cell growth, and the cell began to divide slightly faster than its neighbors. NK cells on patrol detected the abnormal stress markers on the cell's surface and killed it. The cell died by apoptosis, was broken down into fragments, and was cleaned up by macrophages. You will never know this happened. In one of your lymph nodes, a memory B cell that was generated during your last flu shot, or maybe during the actual flu infection you had 3 winters ago, is sitting quietly, doing nothing.
Its receptor is specific for the hemagglutinin protein of the influenza strain you were exposed to. If that strain, or one sufficiently similar, ever enters your body again, this memory B cell will activate within hours, clone itself rapidly, produce high-affinity antibodies, and clear the infection before you develop symptoms. The protection it provides will last for years, possibly decades, and you don't have to think about it, manage it, or even be aware of it. In the tissue of your throat, a few cells were infected by a virus, maybe a rhinovirus, maybe a stray coronavirus that you breathed in earlier today. The infected cells began producing viral proteins, and fragments of those proteins appeared on their MHC class I molecules. But before the virus had time to replicate significantly, the infected cells released interferons that warned their neighbors to lock down. NK cells in the area recognized the infected cells and killed them. The virus particles that were released when the infected cells died were coated by complement proteins and antibodies and were either neutralized or tagged for destruction by macrophages. The infection was extinguished before it ever became an infection in any meaningful sense. All of this happened in the time it took you to watch this video, and this is what your immune system does, all day, every day, for your entire life. A constant, tireless, ongoing process, a living system that never sleeps, never takes a break, never stops monitoring, sampling, evaluating, deciding, acting. There is no peacetime. There is no standing down. The war is always on, and the system is always running. 2 trillion cells, each one independently assessing its immediate environment, each one making decisions based on the molecular signals it receives, each one communicating with its neighbors through chemical messages, cytokines, chemokines, complement fragments, antibodies. No central command, no brain, no plan. Just 2 trillion agents, each following simple rules, and the collective result is a defense system of almost incomprehensible sophistication. The innate immune system provides the fast, broad, violent first response. The macrophages eating bacteria, the neutrophils dumping bleach, the complement system punching holes, the NK cells executing compromised body cells, the interferons warning neighbors. It handles the vast majority of threats without ever involving the adaptive immune system. You will never know about most of the things your innate immune system kills. The adaptive immune system provides the slow, precise, targeted second response. The dendritic cells carrying intelligence to the lymph nodes, the T cells activating and multiplying, the B cells producing antibodies, the killer T cells hunting down infected cells. It handles the threats that the innate system can't contain, and it builds memory, a permanent record of every enemy it has ever fought, ready to be deployed instantly if that enemy returns. Between these 2 systems, the ancient and the modern, the fast and the slow, the broad and the specific, almost every threat is covered. Almost every pathogen is countered. Almost every cancer cell is caught.
almost. Because the system is not perfect. It can't be perfect. The pathogens are evolving just as fast as the immune system, and some of them, HIV, tuberculosis, malaria, the ever-shifting influenza virus, have found ways to stay one step ahead. Cancer cells can evolve evasion mechanisms faster than the immune system can adapt. And sometimes, the system's extraordinary power turns inward, and the very specificity that makes it so effective becomes the engine of autoimmune disease. There is no magic solution to any of these problems. There is no supplement, no superfood, no lifestyle hack that will make your immune system invincible. What there is understanding. Understanding of how the system works, what supports it, and what undermines it. And that understanding leads to a few clear, evidence-based conclusions. Sleep matters, not as a luxury, but as a biological necessity for immune function. 7 to 9 hours a night, consistently, gives your immune system the time and resources it needs to do its maintenance work. Exercise matters. Moderate, regular physical activity supports immune cell circulation, reduces chronic inflammation, and helps maintain T-cell diversity as you age. Not extreme exercise, not punishment, just movement. Most days, for most of your life. Diet matters. Not in the sense of superfoods or immune-boosting supplements, but in the sense of feeding your gut microbiome. Fiber, vegetables, a diversity of plant-based foods. The 40 trillion bacteria in your gut are your immune system's training partners, and you should feed them well. Stress management matters. Chronic stress suppresses adaptive immunity through cortisol, and the connection between psychological state and immune function is real, measurable, and significant. Finding ways to reduce chronic stress, whatever that looks like for you, is an immune health intervention, whether or not it feels like one. Vaccination matters. More than anything else on this list, vaccines are the single most powerful tool humanity has ever developed for harnessing the immune system's capabilities. They work by giving the adaptive immune system the information it needs, without the risk of disease, and the resulting memory cells provide protection that can last a lifetime. Childhood vaccination schedules exist because they work. Tetanus boosters exist because protection wanes over roughly a decade and needs to be refreshed. Flu vaccines exist because the virus changes its coat every year, and your memory cells for last year's strain don't fully recognize this year's. If there is one thing you take away from this entire video about how to support your immune system, let it be this. Get vaccinated according to the recommendations of your doctor, and make sure the children in your life are vaccinated too. And finally, and this is the point I want to leave you with, respect the system. Respect its complexity. Respect the fact that 2 trillion cells are working inside you right now, making decisions you will never be aware of, fighting battles you will never see, dying by the billions in your defense without your knowledge or gratitude. Your immune system doesn't know you exist. It doesn't know your name, your fears, your hopes, or your plans. It is a vast, distributed, autonomous system operating entirely on molecular logic. Self or not self, friend or foe, tolerate or destroy. And it is, without exaggeration, the reason you are alive. Not just today, not just during the last illness you fought off, every single day of your life, since the moment you were born. Your immune system has been the thing standing between you and a world full of organisms that would consume you from the inside out if given the chance. You have never thanked it. You have mostly been unaware of it. You have sometimes actively worked against it, by not sleeping enough, by being too stressed, by taking medications that suppress it without understanding what you are suppressing. But it has never stopped working for you, not for 1 second. not for one day. It has been your silent, invisible guardian for every moment of your existence. And it will continue to be for as long as you are alive. Right now, as you sit here, something is trying to kill you, and something else is already handling it. Thank you for watching. If you found this useful, please consider sharing it with someone who might benefit from understanding how their body protects them.
And if you want to learn more, 2 resources I'd highly recommend. Immune, A Journey into the Mysterious System That Keeps You Alive by Philip Detmer, which is the most accessible and entertaining book on immunology ever written, and the mini medical school lecture series from UCSF, which provides university-level immunology education for free.